Welcome back to Digi Workbook 5, Meiosis edition, Mitosis' more complicated friend. Here we have two really cool gifts that walk us through mitosis and meiosis. So let's watch these and make some observations. On the mitosis side, we start with this cell that has 46 chromosomes. We know that mitosis is only for somatic cells, so this is a somatic cell. And somatic cells are things like your head, shoulders, knees, and toes. Any body part you can think of is made out of somatic cells, with the exception of sperm and egg. Those are the only non-quote somatic or body cells. So here, this guy has 46 chromosomes, and you can see when it replicates, it does mitosis, and it makes a copy, and the copies end up as two identical new cells, also with 46 chromosomes. Let's take a look over at this meiosis side in the green. So here we have one cell. It has 46 chromosomes. It splits. Then we get two cells, and then those split again, and the end result are four different cells with 23 chromosomes. So meiosis is only for gametes. Remember, gametes are sperm and egg. There are 23 chromosomes in the end result, and the last stage, those four new cells, are different from each other. So they're all a little bit different. There's variety. We'll get into that later. Mitosis and meiosis compared. The short version, you know this about mitosis already. Mitosis starts with a cell with 46 chromosomes. Here. It doubles the chromosomes and then it splits in half down here, resulting in two new identical cells, also with 46 chromosomes. Meiosis, however, starts also with a cell with 46 chromosomes. It doubles and splits there, and then it ends up splitting again, and the result is four new 23 chromosome cells, which is half of the original information. And each of these 23s, as I said before, has a mashup of genes from the original 46 chromosomes. So they're not identical to each other, and they're not identical to this parent cell. And this is why gametes need each other to make a complete zygote. They're only half the information, so this could be a sperm or an egg. We'd have four of each for the most part, and that's why they need to combine. So the 23 of the sperm plus the 23 of the egg brings you back to the 46 complete unit of the zygote. We went over this in class, but here it is in case you need it again. We have a DNA recap. Chromatin is a messy bundle of DNA. It's disorganized, and I drew a scribble to represent it, and I drew the scribble by using the line tool up top. I selected scribble down there, and then I found it easier to do it outside of the box and drag it over, but if you'd prefer to do it right in the box, you can do that. I'm just going to get rid of that. And then we have a chromosome. That's a bundle of DNA in an organized X shape right there. If you are a little bit stuck on these terms, go back to Digi Workbook 4 and the video that goes with it. I explain it in further detail there. We have a chromatid, which is half of a chromosome. I used a V to represent half of an X. And then we have the centromere, which holds the two halves of the chromosome together. I used it as an orange dot on here. The orange dot on top of the X. We talked about the centromere cat before, keeping things together. So if you want to paste a little meerkat in there, go for it. Now, we have some new words for things we already know. Diploid. Sounds like an insult, but it's not. A diploid cell has 46 chromosomes, so that's a complete set. That is the same as a somatic cell. So you might hear it said, a cell with a double set of parental chromosomes from the parents. All body cells, or somatic cells, are diploid. And what type of cell division creates those diploid cells? Mitosis. That's what last lesson was about. And here's an image which I'll get to in a moment. Then we have a haploid cell. That's 23 chromosomes. A cell with a single set of parental chromosomes from the parents, or half a set of the complete. And only sperm and egg, which are gametes, are considered haploid. What type of cell division makes haploid cells? Meiosis, which is today's lesson. So, these two pictures. I see four chromosome-looking things. I assume them to be X's, but they kind of look like H's. And then here I see four different rods. But really, if you were to put four sticks in front of you, I'll show you a demo in a moment, and cross them over, you would end up with two X's. So you can see this is actually half of the top. Or if you had 8 sticks and you made them into 4 X's, if you cut that in half, you'd be left with 4 sticks. And here's your demo about it. Here I have 6 chopsticks. So, if I move them around into X shapes, for example, now how many chopsticks do I have? 6. I didn't lose any chopsticks, I just moved them. Now I have 4. And if I rearrange them like that, I still have 4. So 1 chromosome is still equivalent to 2 sticks. And we'll see that in some diagrams. So you might see something like this to represent the genetic material. You might see something like this. That means the same amount of genetic information. A little more practice with the words haploid and diploid. I like this diagram because it really clearly shows you this is one set of information. We use N to represent the number of chromosomes. In humans, this N is 23, but if you look at frogs or fruit flies, they have a different number, so you might see a 4 or a 6 or a 7. Diploid is 2 times that, so we have, instead of 1 red rod, we have 2 red rods. So it's double the red genetic material. 2 purples instead of the 1, and then 2 yellows instead of the 1. So you can clearly see this diploid is a duplicate version of the haploid. So the haploid, cells with only 1 set of chromosomes, 1 gene for each trait, are haploid. We use the N to mean 1 set. All gametes, sex cells, are haploid, and haploid cells are the result of meiosis. H, I like to think of haploid sounds like half. Diploid starts with D, and that sounds like D is for double. Diploid cells have homologous chromosomes, which are the matching buddy chromosomes. There are 2 genes for every trait in a diploid cell, and we symbolize it with the 2N, 2 times 23, in that case for humans would be 46. All of your somatic cells are diploid, and diploid cells are created through mitosis. Remember, D is for double, H is for half. Diploid is double the information of a haploid, which is half of the information. Meiosis, pronounced like my-o-sis. A lot of times you might say meiosis, but it's meiosis. During meiosis, 1 diploid cell goes through 2 divisions to form 4 haploid cells. If you take a look, we have this red and the blue. They double. They start moving around the cell. They end up splitting out. They split out again, and we're back to each of these end cells has half of the information as the first cell, and they're not quite identical. They're a little bit mixed up. There are 2 stages of meiosis, meiosis 1 and meiosis 2. We learned ipmat for mitosis, and now meiosis is going to have ipmat, pmat. Tonight we have a guest star to help you remember how to pronounce meiosis.
My favorite quarantine snacks are Cheerios, SpaghettiOs, and steel cutouts.
Meiosis? How'd you get Meio?
That's not Euro, that's Meiosis.
Euro? But I thought it was Meiosis. We both do meiosis.
Oh.
Oh. Here's our overview in diagrams. So meiosis 1, we can see prophase right there. Our cell is preparing. Metaphase, our chromosomes are starting to move. We'll look at that in more detail in a moment. Anaphase, something's happening where the cells are moving away from each other on both sides. Telophase, we're starting to get 2 new cells. That was ipmat, just like mitosis. Now we go into pmat. In meiosis 2, we have prophase 2, metaphase 2. You can see our chromosomes are lining up in the middle. Now we're going with both cells at the same time. Anaphase 2 and telophase 2, and those will be our 4 new cells. Who cares? Question. We just learned about the neat, tidy process of mitosis. Why do we need this more complicated version? Answer. In meiosis 1, homologous chromosomes swap genetic information, which randomly redistributes genes to daughter cells. This is why all sperm and egg cells are different and why you aren't identical to your parents or siblings. You're kind of like a mashup of their potential genetic information. Swapping genetic information is called crossing over, and you can see here are homologous chromosomes. There are besties. They carry, if it's the gene for eye color, it's on both chromosomes in the same spot. They spend so much time together that they end up switching part of themselves and don't even realize it because they're best friends. Crossing over of homologous chromosomes continued. In Prophase I, the homologous chromosomes line up next to each other. Sister chromatids, remember those are the identical halves, swap genetic information with their neighbor chromosome by crossing arms. The sister chromatids on the chromosome no longer have identical genetic information from their original. So you can see in this diagram, this side was originally identical to its mirror, but when those two crossed over and swapped, now this has a little bit of genetic information from the red chromosome, this has a little bit of genetic information from the blue chromosome. When the chromosomes break apart in Meiosis II, later, the 4 gametes each contain a unique combination of chromosomes. Again, that is why the 4 resulting cells from meiosis are not identical to the original, and it's also why you're not identical to your siblings or your parents. Here's a little analogy for crossing over. Think back to a time where you spent so much time with a friend, a sibling, somebody, where you're not quite sure where you end and where they start. You pick up their slang, they take on your mannerisms, you make the same kinds of jokes, and at some point maybe you even share clothing enough that you're not sure if it belongs to you or them. That's what I like to think about for homologous chromosomes. They are so close together for most of their lives and they're best friends that they end up swapping some genetic material and they don't even notice that it wasn't part of them to begin with. Back to the phases. Interphase. Now, there's only one interphase for meiosis, which we will explore soon. But just like interphase of mitosis, this is where we duplicate the chromatin, so we copy the DNA to prepare for Prophase I. Here's a nice diagram that someone drew that I found on Google with everything labeled. In here, our nucleolus is inside the nucleus. We have our chromatin, we have our centrioles getting ready. Our nuclear envelope is the membrane around the nucleus, and our membrane around the outside. So, interphase, just like mitosis, in meiosis, we're going to copy the DNA. That's the most important thing. Now, the phases of meiosis are labeled 1 and 2. So we go interphase, prophase, metaphase, anaphase, telophase, 1. Then we go prophase 2, metaphase 2, anaphase 2, telophase 2. So the 1s and the 2s, we go through all the 1s before we get to the 2s, but there's no number after interphase, which we'll talk about soon. So prophase 1. The chromosomes condense, just like in mitosis. The nuclear envelope, meaning the membrane around the nucleus, goes away, breaks down, just like in mitosis. But this time, our homologous chromosomes are going to pair up. So they're going to trade their socks, just like we saw in this diagram. They're going to swap genetic information during prophase 1. Here's an image where you can see the centrioles are moving around to the other side of the cell to get ready, and here it doesn't move, but you can see those chromosomes have now intertwined, the blue and the green, and that's the crossing over part. They'll separate in a little while. Different from mitosis, the chromatids in meiosis are going to exchange their genetic material when they cross over. Recap, write down here what are homologous chromosomes, write your answer there. And on the picture, either the top one or the bottom, there are homologous chromosomes featured in both. Go ahead and drag this arrow to point them out so that I know you know where to find them. Metaphase 1. In metaphase 1, the spindle fibers move the pairs of homologous chromosomes to the middle of the cell. The paired homologous chromosomes stay paired. So you can see here they still have their buddies. And unlike in mitosis where there's one line of chromosomes, a single file line, in meiosis they bring their buddies with them. Here we have both of the chromosomes in the pairs. They're still together and they're lined up in the middle of the cell. If you'd rather look at this diagram like this, that's fine too. Just remember cells are spheres just like the Earth. So up versus down is just what we as humans like to think of as the perspective of the cell. But really, it could be any direction in your body. So, compare this to mitosis. Write down here a little blurb about how metaphase 1 of meiosis is similar or different or both from mitosis. If you need to look back, we have images of mitosis in Workbook 4, or you can Google it. Next, we have Anaphase 1. Homologous chromosomes separate from each other, but they're still whole chromosomes. So you can see here, it's kind of weird because it looks like the chromosome's being dragged from its middle and then it's folding in on itself. There are 4 little legs on there. Those would still be an X shape if you flatten them out. So there's a whole X being dragged from each of these to both sides. If you think back to anaphase of mitosis, it was just a chromatid. It was just a V, half an X being dragged. So we still are moving around double the information in this cell. Same mechanism as mitosis. The spindle fibers are pulling on the chromosome and they're pulling them to the opposite poles or ends of the cell. What to notice, the chromatids do not separate at their centromeres this time. Each chromosome is still made up of 2 chromatids that are attached to the centromere for now. Dun dun dun. Telophase 1. During telophase 1, the cytoplasm divides, cytokinesis, just like in mitosis, and 2 new cells are formed. However, we're not done yet. Both cells have 1 chromosome from each pair of homologous chromosomes. So we separated the buddies there. So there's still whole chromosomes moving to both sides of this cell. And now we're going to divide again. But wait, there is only 1 interphase. Question, why does that make sense? Hint, what happens in interphase and what is the end result of meiosis?
You are a human. And as a human, you have 46 chromosomes in all of your body cells. Now, if you're going to make some new chromosomes, you're going to double your DNA. And for a short time, you're actually going to have 92 chromosomes. But, as you know from mitosis, you're going to split that in half. You're going to end up with 2 new cells, each with 46 chromosomes, identical to the first one. Now let's say we're doing meiosis. So we did the same thing. We started with 46 chromosomes. We doubled it. We had 92. We split that out to 46. But the end result of meiosis is 4 haploid cells. So this 46 splits into 2 23s. And this 46 also splits into 2 23s. So that's how we started with 46. We copied it. This is interphase, where we copy the DNA. Mitosis, we'd be done here. But if we're doing meiosis, we go all the way down here to the 4 new cells. Now, the processes are a little more complicated than that, which we're going to go through in these lessons. But if we doubled our DNA here and we had 92, do we want to do that again down here and have another interphase if we're going on to meiosis? No, that would end up with, if we had our 92, we went to our 246s, each of those doubled again, so we'd be at 92 and 92. And then if we split those out, we'd end up with, 4, sorry, getting a little messy here, that's a 46. 4 46s, which, if you're a sperm or an egg cell, and you have 46 chromosomes, you're not going to have any room for the other gamete to make that 46-celled zygote. So this is why we don't do more than 1 interphase. We only want 1 interphase. Otherwise, we would have too much genetic information.
Okay, try your answer now that you've watched that. Remember the question, why does it make sense there's only 1 interphase in all of meiosis 1 and 2? Answer it there. Meiosis 2 is most similar to mitosis because the 2 daughter cells from meiosis 1 divide again. We'll look at that with some diagrams so you can figure out what that sentence means. By the end of meiosis 2, there are 4 haploid daughter cells. So now we're moving into prophase 2. You're going to see Roman numerals. Depending on the font, it can be hard to tell what it looks like. So I put this 2 in parentheses for you. But if you see something after any of the phases of mitosis, it's going to be a 1 or a 2, even though it'll look like I or II. In Prophase II, new spindles form around the chromosomes, and now we're going to have 2 cells going at the same time. So it's just like mitosis. Now we're going to go PMAT. So we're preparing during prophase. Metaphase II, the chromosomes line up along the equator, the middle of the cell, and they're attached at their centromeres to the spindle fibers. Now look, here we have a whole chromosome lined up down the middle. Looks just like mitosis metaphase. This is Metaphase II of meiosis. For reference, here's Metaphase I of meiosis. So take a look there. What's the difference between this diagram and these 2 diagrams, aside from the orientation of the sphere? Right that there. What is the difference between Metaphase I and Metaphase II of meiosis? Down there. And say that 10 times fast. Anaphase II. In Anaphase II, the centromeres divide. The chromatids move to opposite poles of the cell. Remember, this looks exactly like anaphase of mitosis now, but we're going at the same time in 2 cells. So the chromatids move to opposite poles, opposite ends of the cell, and think back, what is the difference between Anaphase I and Anaphase II of meiosis? So we're looking at Anaphase II right now. Scroll back up to Anaphase I and tell me what you see. Here's Anaphase I. Here's Anaphase II. Next we have Telophase II. During Telophase II, a nuclear envelope or membrane forms around each set of chromosomes, so that's what this dotted line is coming back. The spindles break down, go back to where they came from. The cell goes through cytokinesis, so it's going to split the cytoplasm, and the end result of meiosis is 4 haploid daughter cells. So 1, 2, 3, 4, just like mitosis. Ta-da! And in a human, a haploid cell, as you know, has 23 chromosomes. And we just made either 4 eggs or 4 sperm cells. We made gametes. So let's add, these are gametes. And it's very exciting, so we're going to highlight it so that you don't forget that fact. There we go. Great. What could go wrong? Well, for one example, nondisjunction, which is non-disjunction. Non means not, dis means anti, junction means come together. So this is not come together? What? This means they're supposed to separate and they didn't. If homologous chromosomes don't separate when they're supposed to in anaphase I, an extra chromosome is going to get dragged around throughout all of meiosis II. This means one gamete will have an extra chromosome. This can lead to a variety of genetic disorders, including Down syndrome. So if you look at this diagram, let's say these spindle fibers don't attach to the centromere of that chromosome where they're supposed to. That means this whole chromosome is going to get dragged to this side. These 2 will still split as they're supposed to. And then you'll end up, this gamete's missing a chromosome, which also leads to genetic disorders. This gamete has an extra chromosome, which can lead to genetic disorders. Now, sometimes you can have a missing chromosome or an extra chromosome with no side effects or genetic disorders. But more often than not, if you don't have the 23 chromosomes you're supposed to have and no more or no less, it typically is a genetic disorder. Okay, we've seen this before. Now you're going to draw the chromosomes in the cell as it undergoes meiosis. So we're going to start with the parent cell, and guess what? I forgot interphase on this diagram. So you're going to add interphase with a circle. I got that from the shapes up top. And we're going to need a nucleus inside this circle. So here's a little circle. And we're going to need some chromatin inside this nucleus. So we're going to give it a little scribble. And we should label it because we are good scientists. Here we go. That is interphase. And guess what? There's only one interphase, as you wrote about before. So you only need that one. And then remember, it goes interphase, pro 1, meta 1, ana 1, telo 1. This diagram goes this direction. Prophase II, here are 2 cells that are doing prophase II. Metaphase II, 2 cells doing metaphase II. Anaphase II, 2 cells doing anaphase II. Telophase II, since they're starting to split, you have this is going to be 1, this is going to be 2, this is going to be 3, this is going to be 4, and then 4 daughter cells. Remember, they are not going to be identical to interphase, and they're not going to be identical to each other. So if you want to get creative and add colors, that would be great. It will really show that I can see that you understand this. If it's too complicated, that's okay. If you also want to do this on paper, I'll accept it. Or like last time where you take a screenshot and draw on it in Snapchat or Notability or a different app where you can draw on it, that's all fine. And here I have my diagram handwritten if you want to look at it. We have interphase, prophase. You'll see that this nucleus has 1, 2, 3, 4, 5, 6, 7, 8 chromosomes in it. That is our duplicated set. So we see 8 whole chromosomes moving around the cells. And then you can see 4 by the time we get to our first telophase. Same with prophase. And then they're lined up single file by metaphase. They start to split out during anaphase. And then if you added 2 V's together, it would be the shape of 1X. So this is really just 2 X's, 2 chromosomes in every end result. You can suspend your disbelief. This diagram shows you duplicated genetic information. So our original cell would have had 4 chromosomes in it, which is how we got to the 2 in these daughter cells. And our prophase 1, where we have a full copy of our DNA, is showing 8. This is as if we had 46 in our original cells as a human. If you drew all 92 replicated chromosomes in there, and then you traced 92 chromosomes as they move throughout the cell, by this point you're now down to 46 in both cells. And since we want to get back to our 4 haploid daughter cells, here where they split, then you'd have 23 V's. 23, 23, 23, 23, 23, 23, you get it. Here are some fun facts that you'll need to know about this diagram. The little V's represent chromatids. The little cylinder tic-tacs are centrioles. The lines are spindle fibers. The X's are chromosomes. And for most important facts that I could think of when I drew this picture, besides IPMAT, PMAT for meiosis, it produces 4 gametes, which are sex cells like sperm and egg. 23 chromosomes in a human is a haploid, a half set. All 4 daughter cells are a little bit different. And this happens in the gonads, the ovaries or testes, which we haven't really talked about yet. All right, so scroll back up if you need help on anything. And your homework is to finish this slide. Good work.
All right. My favorite quarantine snacks include Cheerios.
Meiosis!
Okay. All right.
Yeah.
My favorite quarantine snacks include Cheerios, SpaghettiOs, oatmeal raisin cookies, and recording for her class.
Alright, a little yowling.
All cut out.
Meiosis? How'd you get
No.
my O?
That's not your O, that's meiosis.
No, it's meiosis. We both do meiosis.